Luc, mute le canal 1 s'il te plaît.
Hello, everyone. Welcome to this session on AI discovery, drug discovery with AI. It is the last session of this very packed AI week. So I'm sure the excellent technical team must be exhausted. So thank you in advance, guys, for the support. And thank you, Nathanael, and all the AI for Belgium team for the organization of this week. So I'm Thibaut Halleput. I will be the chair for this session. We have a very nice panel of speakers today. And I will make the link with the general topic of this AI week, which is sovereignty. One thing that is required to be sovereign in any given topic is to dispose of the necessary resources. And you will see that in Belgium, we can be proud of the ecosystem that we have regarding healthcare in general and drug discovery in particular. And so you will see that the speakers have a very high level, each in his own position. Maybe as this session was positioned as an intermediate level session, I will maybe briefly remind some basic but nevertheless important concepts related to drug discovery. So what it is, this topic of drug discovery? Well, it starts from the identification of an indication, so a pathology, a disease. Then we try to understand what does this disease or this pathology to the organism that it targets, so to the human body in particular. And then once we understand that, we can start thinking about new ways of curing the disease or controlling it. So to give very practical examples, if you think about rheumatoid arthritis, for example, which is a very common disease targeting almost 1% of the population, researchers, clinicians, doctors try to discover the symptoms, the phenomenon through which the disease produce manifestations. So in the case of rheumatoid arthritis, it will be bone degradation, inflammation, etc. So researchers try to understand that. And after some months, some years, some decades, sometimes they can come with a more precise understanding of how the disease works. You see here on the right, still for rheumatoid arthritis, all the symptoms, so inflammation, bone destruction, angiogenesis, etc. But now we have a more clear understanding of the metabolic pathways, the mechanism through which the disease affects or changes the organism, so the body. And once we understand that, we can start thinking about targets, so elements of these mechanisms, of these strange behaviors induced by the disease that we can target to cure the disease. And so this is target identification. In the case of rheumatoid arthritis, it would be, for example, the TNF pathway. And the TNF, so it's a kind of messenger in the metabolism of humans. And then we can think about drugs that we can design to capture or to destroy or to limit the effect of the dysregulation that was induced by the disease. And you see here concrete examples of 4 different treatments, 4 different drugs invented to capture TNF to regulate the disorder induced by rheumatoid arthritis. And they have each their own way of being produced, and they have their own mechanism of action. But they target the same target. So you see, we have one indication, multiple possible targets, and for each target, we can think about multiple ways of addressing it. Sometimes we can also think about repurposing an existing drug targeting a given target, but for another disease, and this is called drug repurposing. So all what I've just covered, we have been doing that for decades now, coming with new drugs for a lot of different diseases. Another question that will be addressed today is how can we change the way we have worked so far thanks to new AI and data-driven tools. And that is what will be covered by the speakers today. So later this afternoon, we will have the chance to welcome Dirk Ramakers, the chair of the FPS Health. He will join a bit later. And I was mentioning earlier that we have a very good ecosystem here in Belgium with respect to drug discovery and healthcare in general, but also for this AI-related innovation. And we propose to organize the rest of this session in the order that is displayed now on the screen. And I was also referring to the different components required to reach sovereignty for a given topic. And you will see here that we have representatives from academia, namely with the presence of colleagues of VIB. And then we will have, slightly later in the maturity chain, we will say, a startup incubator represented by Aquila Bioventures. Then a nice example of startup active in this field with Cantify. And then we will have also the chance to welcome a representative of the corporate pharmaceutical industry with the presence of a colleague of J&J. And then also, last but not least, we also have foundations active in this field, sometimes putting the stress on topics that are less regarded by other players, and that is also very important. And for a testimonial around this matter, we will have the chance to welcome Fondation Sang et Génome here on stage. So we will organize the rest of the session in a succession of talks of around 15 minutes. And then we will have the opportunity for the audience here, but also online, to ask questions. I know that there are more than 100 people registered to attend this session online, so likely a lot of questions there too. Technical modality. You see that what I say is transcribed in French and in Flemish. I will ask the questions also to be asked in English, if possible, because the translator requires English as an input. And now, that being said, I will welcome Eric Verschuren from VIB, so more an academic but also translational research institute, very well known, to share his views on AI-driven drug discovery. So, welcome, Eric. the PowerPoint. Yeah.
Thank you for the very nice introduction. Explain the drug discovery process already saves me well a bit of time during my talk. So it's a pleasure to be here today. Like in the introduction was said, I'm Eric. I'm leading the AI studio at the VIB, and more specifically at the VIB Innovation and Business Department. And today I'll argue where AI is already operational in the drug discovery pipeline, but also I'll try to highlight with two examples where it actually still fails, meaning where it goes out of distribution and where results are imperfect, and how we can actually try to solve that. And I'll try to also make the link at why Europe's advantage in sovereignty and what we need to focus on in Europe actually runs through data for AI. Let me introduce VIB as a starter. So VIB is the Flemish Institute for Biotechnology. It's really a scientific powerhouse already since 1996. At VIB, we have over 2,000 scientists that are distributed over 9 research centers, active in like most important areas of biology and disease, like cancer research, neurobiology, plant systems biology, inflammation research, and so on. These scientists are actually co-affiliated at the 4 major universities in Flanders, being the University of Ghent, Leuven, Brussels, and Antwerp. Our scientists are overall excellent. They're also actually selected on their excellence, and that's really reflected in how highly they are cited across the world and in the number of European Research Council grants they manage to really land each year. But also at VIB, we really believe very strongly in that excellent science is often propelled by excellent technology. So we also invest very heavily in a rich ecosystem of core facilities that really support the science to generate the excellent data that they need to do their excellent research. But also VIB is not only about academic science, it's also really about translating that science into value, into impact. And I think just, like looking at some of the stats here, it speaks for itself. We are very good in protecting our IP. We rank 7th in IP filings. We have a really outsized business development department, which goes out and finds these collaborative partnerships with local but also worldwide industry. And this is also really important because this is really where the rubber meets the road. No, we can have great scientific ideas, but we also are very keen to translate things to impact, and that impact is usually done in industry, right? In small to biotech to large pharma. We're also really excellent at founding new ventures. And so actually we rank 7th in the European Research Center spin-out ranking. And that's something that we're obviously very proud of. So like I said, we have an outsized innovation and business department at VIB, which is composed sort of these elements that I'm displaying here on the tile. We have our IP group, we have a new ventures group, a business development group, and then we have 2 teams that really bridge the academic science towards the valorization. That's both on the experimental sides to the discovery sciences team, and also recently, since last year, a new team that we founded, the AI Studio, that does the very same mission, but for digital assets. So think new AI models, finding valorization paths for them, but also really finding valorization paths for the rich data that's being generated at VIB. So again, coming back to also what was already hinted to in the introduction, AI really impacts drug discovery end to end. So it goes from target ID to drug discovery, preclinical studies, clinical trials, all the way to approval, launch, and post-market surveillance. But I think what I will try to argue is that the maturity and how it's impacting each of these phases is still uneven, and obviously there's still work to do here. And I will make that argument by speaking about 2 examples of 2 really projects that were actually positioning squarely onto a valorization path at VIB. Obviously, both projects, both cases are very much at the front of this drug discovery pipeline. That's really where VIB is most active. And target identification and drug discovery, preclinical and clinical is typically more the domain of pharma. But first, before I go into the 2 cases, let's take a small step back and look a bit at a higher view. Why is molecular biology such a hard problem to solve for AI? Because we've seen such great advances in how the AI models do on text, images, for everything, for really there is rich data. And that's really the point, right? We have enormous amounts of text. Text is cheap to generate. Text is massive. We have a whole internet full of text. But on the flip side, for biology, we don't have that. Generating biological data is it's expensive. It takes a long time, so it's slow, and it's also sparse. For example, we have only 1 billion of sequences for less than 0.10% of the natural sequence space. Generating that data really requires costly wet lab experiments, and even then, it's still sparse. And most of all, that data is private. You don't find that data like spread out all over the internet. Secondly, and this is important second difference as well, when you type something in ChatGPT, you get immediate feedback. If it's not perfect, it's also not a big deal. You just reprompt and you move on. You don't have that same luxury when you're dealing about biology AI. The feedback is hugely delayed. You get your AI to propose an experiment, then you still have to execute it. That feedback is delayed by weeks and months, but that's not the only problem. When it's imperfect, the impact of the mistakes is also huge. These imperfect predictions, they can delay drug programs by months or even years, and they burn millions in resources. It's not just a reprompt. So I think if you want to really define like what drug discovery AI performance looks like, it's not only a function of having the biggest model, having the largest compute, having diverse data. That data, well, those are all true, obviously, for biology AI models as well. Generating that biological data with that diversity is just really hard, and you need data that's real to what you want to predict, and that's the second problem. And just don't take my word for it, I will quickly walk you through two really recent examples, and they're that recent that they're really only like 6 months apart. What you see on the left here is an independent benchmark of, let's say, the largest foundation models that are applied to predict basically the effect of perturbing a gene for a disease. And much to, I think, everyone's dismay, we found that the performance of the foundation models, so the middle block there, and the deep learning models, was really no better than just doing a simple linear model with additive terms. After all these resources, this is where you land. That's shocking, right? But obviously, like, things are moving really fast, and only like earlier this week, so I had to adjust my slide. I have a really heavy US bankrolled company called Zara, and I was fortunate to work there. Like, dropped the latest, biggest foundation model for biology called XL Ultra. It now has like 5 billion parameters, and it's also trained on the largest perturbational single cell data up to date, about 25.6 million data points. And now they're claiming that we're starting to see the scaling laws that we talk so much about in AI also to be applicable for biology. But I think it's still a far way to go, and the jury's out, and obviously this still needs to be independently validated. So I guess this is really also where I want to point a little bit, like, where are we in Europe? We don't have the luxury to throwing billions of euros at startups. That's just not really our game. But what we can do is be smart about like how we approach our model building, and also be smart about our data. And that's really how I think we envisioned at VIB sort of the thread through all the groups that are forming our newest center, which is the Center for AI and Computational Biology. And we really want to look at this problem of AI for biology at a holistic scale, if you want. We have all these scales, like basic biology starts at the genome and then goes to the regulatory level, and then these, the regulated mRNA gets translated into proteins. These interact into a cellular context, in tissues, in organs, at a population level, and so forth and so forth. So really what we want to do is we want to model these contexts in a bridged way. We want to pair increasingly hierarchical through this context, and we want to generate the data that we need to train the models that can bridge these contexts. And obviously that's where it's great to be at VIB, because we actually have that whole rich ecosystem of generating data. We have all these excellent research groups that are active in plant system biology, in microbiology, in neurodegenerative diseases, in cancer research, and so on. So it's really, I think, the marriage of finding the best model that can really grasp this complexity across the multiple scales, and generating the data that you need to train these models. So that's really our mission. A few examples, as I said. So this concept of bridging scales and generating the data that you need to bridge these scales to train your AI is really something that we're super excited about, and that's something that we're actually positioning as a new venture idea. I can't really go into the details here because we're a few weeks away from filing the paper and filing the patent, but just to say that at the heart of it, that is the concept. We're training these deep learning sequence to function models that we are going to position towards target ID to find and understand disease cell states and how to turn them back into healthy. But also at the same time, we can use these models, or we aim to use these models to design gene therapies. And so how do we now fix these disease cell states? And also to say for which patients are these therapy actually going to work in the best way, and that we can do that through whole genome variational data. And secondly, this is my second example from the drug discovery pipeline. It's around designing molecules with AI, right? And this is, I think, the area where we've seen a few years ago already now the most impactful example with the rise of AlphaFold and then diffusion models. And I don't know if you remember, but that was actually the subject of the Nobel Prize 2 years ago. And they work great on generating like novel proteins, on predicting the structure of proteins from sequence, and it was a huge leap. But again, as the point that I was trying to make, maturity is uneven. And if we see at this community benchmark that was actually done a few months ago, and that was really exciting, they break out of distribution problems. The subject of that benchmark is like, can we design a therapeutic grade binder, a novel protein against the spike protein of Nipah virus. It's a huge problem. There's no cure for Nipah virus. It's an important pathogen identified by the World Health Organization, and it's out of distribution. No structure, no precedent on binders. So perfect for a unbiased benchmark. And so many groups contributed actually in this challenge, which was super exciting to see. Came up with a bunch of different formats, both mini proteins, both antibodies, both nanobodies. About 20,000 synthetic sequences were designed and also tested, benchmarked in the same experimental platform. But then what we saw is that success was uneven. So for these mini proteins, these are these small helical bundles, very abundant in the PDB. Success was great. We could get many, many mini binders binding against the Nipah spike protein, but we couldn't get any therapeutic proteins to bind. There was only one nanobody that bound with really poor affinity. So that's a huge problem, because in the whole drug discovery pipeline, antibodies are the workhorse. We still haven't put mini proteins in people, and we're also not planning to do that anytime soon. So can we get it to work for antibodies? And also, why does it not work for antibodies? And that's really, it's all, can all be in the same way we reduce to the out-of-distribution problem. So again, this was actually the point that I was making on the previous slide. These large models, I think protein LLMs or these structural diffusion models like RF diffusion and BOLTs, they're trained on the PDB. What they can predict is proteins that look like proteins in the PDB, but they break on things that are not in there, like the antibodies. So our answer or solution at VIB for that is to say, okay, if we don't have the data, maybe we need to go back to what we know that we can do well, and we're actually trying to enhance now our AI with good old first principle biophysics by infusing that back into the AI, as to really ground these models into first principles. That's not the only thing. You just don't want binders. You want binders with therapeutic grade quality, meaning they have to be stable, they have to be soluble, they have to be non-aggregation prone, they have to be non-immunogenic. That data is also not available. That data is actually, no offense, locked away at the big pharmas and the biotechs of the world. We don't have access to that to train our own models. So what can we do? We can work with what we have, which is limited experimental data. But we can bootstrap our way into this problem. So at VIB, there's a lab actually where I originated my scientific career, the Switch lab, that together with the Serrano lab came up with what we call like a first principle biophysics force field called Foldix. And you can use that force field to really generate synthetic data. Now, it would be easy to say, like, let's use that synthetic data to also train our AI, which is probably not a good idea. But what we can do is we can use that synthetic data to say which are the experiments that we need to do to better train our AI, to really help identify us where the gaps are. And that's typically what we call these lab-in-the-loop type experiments. And I think that's really where the promise is to train these type of models, so that we can generate synthetic data or have the AI really like infer the data that is needed to really improve on its own success. It needs to be diversified, right, and on the right properties. So again, what this buys you is you can get away with smaller data, but you still need a model that can do really well on smaller data as well. And I think that's also where the group that developed this concept, these platforms, came up with a really neat trick. So they developed an algorithm called MADRAX. And basically, MADRAX does it can constrain your neural network during training. So what you do is you don't have to explore the entire space. You basically explore the space constrained by biophysics. And I think that's a really neat trick to say, we have the data that we have, now let's train like the model that is really good at doing this with the limited data. So again, you can do this cycle over and over again, and I think that's really where the promise lies to really get at these generative AI models to do well on antibody designs as well, as good as they do on protein designs. Obviously, to do all this really well, it takes a tight coupled ecosystem to turn biological and translational data into AI with real drug discovery impact. And that's something that, again, I think at VIB we have done really well. So we managed to put like what I call the holy trinity together in a way that it's supportive. So you need a secure, fair data backbone. We have an excellent data core that can really govern our data, that can make sure this data is reusable, that it can be exposed in a federated and compliant way, and it also gives us the compute infrastructure. We have an excellent machine learning unit, which is being expanded at the moment as well, that can really think about like the new class of models that we need to develop that need to be biology aware, that need to be explainable, that you can really use confidently in other distribution contexts. And finally, and I think this is like really, really key here, we have an integrated wet lab technology that we're building as well, where we can really generate like ML grade data. And again, like this is it's not just about taking all the data and putting it together, it's about generating that data in a deliberate way, that you know where the gaps are in your model and that you can generate the data very deliberately to fill those in. And with that, I'll come at my closing slide. So, when we think in Europe about sovereignty, we always talk about the compute, the models, the data, the regulation, the talent. To me, in my opinion, I think where we really have to pay attention to and where we can really win compared to the other like geopolitical powers in the world it's on data. Data is the bottleneck. Without the right data, you just cannot go out of distribution. So that's our strength. I think we do this already really well in Europe. Obviously, we can do better, and we can make sure that between institutes and even maybe industry, our data is shared, not just shared in an open way, but in a way that is federated. Obviously, there's important constraints around that. So I think, we have our advantage in Europe. Let's just make sure that we really capitalize on it maximally.
Okay, thank you for this very interesting presentation. Are there questions in the audience? No. Yes?
Could you say one or two words about, you had the synthesis lab mentioned there. Do you know, and can you share a bit about what's the status in here about synthesis prediction, synthesis automation?
Yeah, I think we're building this unit still. Like it's in full building mode, but I think it goes through robotics to plate-based assays to make sure that you have cellular systems that are very close to the diseases that you want to model, that you can perturb in the right way and at scale. So it goes into like as you can imagine, right, inducible cell lines, robotics, fast turnarounds, readouts at a single cell. That's really how you want to generate your data.
Yeah. This is a kind of game. I will throw the mic.
All right. So congratulations for your presentation. So you say you're hiring. Could you tell us about the process of hiring, your requirements, or what are you looking for?
I don't know if you saw, but you can just scan the QR code or drop us an email.
Okay. All right.
Yeah.
Okay.
Yeah, so we're building the AI studio with a variety of
tools,
profiles.
right?
Yeah, right.
Okay.
Yeah. Both on basic ML as applied ML for target ID and drug design.
You can throw it over there. Yeah.
Thank you, Eric, for a very nice presentation. It's good to see that Switch is still mentioned on your slides. Yeah. It's very good.
Back to basics.
Back to basics, yes. You pointed with yourself, there's a lot of hype being about AlphaFold, but you as I know that there's a lot of problems also with this AlphaFold. So to what extent are you guys looking into the dynamics of the proteins? I mean, the problem is that proteins are not static structures, they're flexible things. Yeah. We looked into allosterine and all this kind of stuff in the past when we were in Switch. Is there any way, or can you tell me maybe what the status is in that context, and if these things are very far from dealing with that, or if they're actually approaching it in a proper manner?
Yeah, that's an excellent question. I think if we're talking about like lack of data, if we grossly lack of dynamics data. The lab of Charlotte Dean actually is making huge effort in trying to collect or at least standardize all the molecular dynamics data in the world so that we can train models that can also do dynamics. It's one of those next frontiers that if you talk to the DeepMinds and the Zaras of the world, that's where everyone wants to go next, but there's still a huge amount of data that we need to do that. I think that said, it is also a data problem, but that said, I think, you're also not constrained to just test one binder. And I feel if we can test a diverse enough set of synthetic sequences, whether they bind your interface, there's a certain amount of luck, which you can obviously put the stakes in its favor. But if something binds, you have something that you can go on. You don't necessarily need to fully understand the dynamics of it to get to a therapeutic protein, fortunately. So.
Another question? Thank you.
Thanks. Doesn't work. Yeah. Can you hear me?
Yeah.
I was wondering, you worked at Xaria, and what are the lessons and know-hows that you could take from those experiences and translate them into the VIB experience, and then how did that help you?
Great question. Well, I can't obviously disclose everything there, but it's pretty much what I incorporated in my talk, right? When we started at Xaria, we were super excited about the antibodies that were generated by through Rosetta Fold diffusion that came out of the Baker Lab, which was really the seed that started Xaria. But on the first real targets that we tried it on, so not the targets that are overpopulated in the PDB, like the PD-1s, PD-L1s of the world, but something that really still matters, for which we don't have any therapeutics, it just didn't work. And that was a first sort of cold realization. And then secondly, yeah, honestly, the second realization was also what I had in my talk, is that the very first model that we trained, and it was actually, I think, the second bar from the top there, XL on only a fraction of the parameters and a fraction of the data, did not do any better than just the simple linear model. So then, yeah, I think they went full force into generating that data that you need to do better on the biology inference models. Yeah.
One more question from my side. So we see that the language model for human natural language evolved quite fast. And to be a bit provocative, do you think that you could just wait a few months that foundation model for proteins will get so good that you will have the solutions that you are looking for? Or do you think that you have a specific role in that evolution?
Yeah, it's also a great question. And obviously, the size and the number of parameters of the model matters, but I think it's really not going to do well still without the right data that you need to get those models to perform well out of distribution. So yes, it will help. So yes, it will make an impact, but only if we generate the right data with it. Because otherwise, you're just going to learn how to do bias predictions even better. So.
Thank you. Thanks once again. Now we will welcome Florence Bosco from Aquila BioVentures.
So, hello everybody. Thanks for having me here. Very happy to be here. So what I'm going to talk about today is giving you the feedback from an entrepreneur who decided to put his own saving and convince the people around herself who trust to also invest money in AI in drug discovery. So I'm in this venture now since 2 years. We are starting to reach the first milestones, and we are still there and still convinced we should have done it. So that's quite good news. So what we are building is Aquila BioVenture. It's a venture creation firm that is enabled by discovery platform that is maximizing the use of AI at all levels. So we create tech bioventures from causal target discovery. So the venture builder model was important to us. Why? Because we didn't want to only make a one shot, just try AI and that's it. We really want to demonstrate that AI is changing the game in drug discovery, really decreasing costs, time, and improving the success rates. And we wanted to repeat that in different therapeutics areas. So that's why we went for the venture builder model. So the model of Aquila is this platform first. It's Aquila Brain, which is combining the best patient-derived disease models, then causal biomachine learning and chem AI algorithms, and enabling the discoveries that we are making. And then we go into this process of discover, build, and realize, so exit the spin goes towards investors or pharma players. But maybe a very good link with the previous speaker, of course, Eric, and thanks a lot for your insights. When I started this venture, I thought, can I find people, knowledgeable people out there that will join in this initiative from different perspectives? And then the first person was Reginald Brice, who you can say is an elite drug hunter. He was the former VP drug discovery at Galapagos. And is now part-time head of discovery for us. So he has a huge experience in target discovery, having worked with about more than 100 targets in his life. And then the second very important person is Stijn Arts, that was mentioned by Eric before. So Stijn is the scientific director of VIB.ai, and I call him a biomachine learning superstar. So I was very lucky to meet him, and he accepted to start a collaboration with us about target discovery. So and we are like the beta tester, the early adopters of what he is developing in terms of causal biomachine learning in target discovery. And then the last person, he is business angel from the pharma industry, Laurent van Lerberg, who joined and invested in the company. And so together with these three persons on board, I thought, well, this is really worth giving it a try. So now we raised 2 million so far, and we are in our first project, reaching the first milestone, which is the in silico target nomination milestone, so very important. So what is the pain? So let's take a few steps back now. What is the pain we are answering? So there is a big fact in the industry, which is that 70% of the pharma R&D pipeline is based on herded targets. So what is herded targets? What is a herded target? It's a target with more than 10 drugs in development. So if you look at the druggable genome, this is a view from the Illuminating the Druggable Genome Consortium, where they classified the targets. So in theory, we have more than 20,000 targets that are available in the genome, and only 10% of them, so the pharma R&D pipeline is focusing on only 10% of the druggable genome. And there are still a big percentage of targets that are called dark, so where we have no knowledge at all, and more than 50% of targets where we have some understanding of the biology in which they are involved, but no drug in development. So this target herding is creating is producing incremental assets that are characterized by, a poor differentiation. So it's only optimization-based innovation. Also, then you are entering into crowded therapeutic markets, and you are under pricing and competitive pressure. But the most important thing is that you are bringing limited patient outcome improvements. So the current model in the biopharma industry is really compressing differentiation, and pharma industry is scouting for better targets and novel biology. So this is what we want to address, and then what is needed to address that? Well, you really need disruptive approach to novel target discovery. And there are a few things you need to do really differently. First, you need to explore biology upfront in a very massive way and hypothesis-free, to move away from the one hypothesis at a time way of doing target discovery, really explore massively biology when it's cheap to do it very early on. Then the second point is you have to increase predictivity of your models, and have models that are scalable, where you can produce enough data. And the third point is definitely you also need to expand the actionable chemical space. And so these are the three key things where we need to adopt disruptive approaches. And there are a lot of tools that have been developed now, a lot of technologies. And what we have done, so we have engineered this discovery platform called Aquila Brain, where we have combined and engineered two lab-in-the-loops. So we have two loops. It's also the lab-in-the-loop approach that Eric mentioned. So basically, you generate wet lab experiments. You perform wet lab experiments, you generate data, you feed your algorithms, they produce hypotheses that you test again in your wet lab setting, and you can loop again, and you in this way you optimize the cost and time to run this this target discovery phase and reach much better informed results. So we have two loops like this, one for novel target discovery and one for de novo molecule design, which is running the same way but with different tools, of course. So the approach is really the lean startup approach, really going out there in the world, looking at what are the best tools, what are the best technologies that have been developed so far. And for the first space where we did that work of deciding which technology we would like to use was in the how to explore biology upfront. So how does that happen now? So mainly, with the progress of sequencing, we have new data modality, we have now big data, and then some techniques arise from there. So first of all, genome-wide association studies, where the first studies that were designed to identify new targets. And from there also differential gene expression studies, which added a layer of gene expression, not only mutation and variance, but also gene expression, which was adding a layer of precision. However, we are still there in correlation, association, not in the causality space. So you see the cat there, well, he didn't cause the crack in the roof there, but he is located there in the crack. So then a very interesting technology appeared to us, and this technology is a gene regulatory network inference. So this is very interesting because it brings you really closer to causations, to understanding causality. So with gene regulatory network inference, the disease is modeled as network state, and you can compare two network states. And you can do in silico perturbations, and you can find tractable intervention, actionable interventions. So, and the leading, well, we were speaking about sovereignty, and what was interesting is that once we started to focus on GRN, it became very clear that we had the leader here in Belgium with Steinhardt. He was always coming on top of every publication, benchmarking, and so on in that field. So, so this is something where we are really ahead of the field in Belgium. So that's the first. So this bio machine learning causal approach is enabling really to systematically explore disease causality upfront in silico when the cost is low. And it's an important early de-risking, although it's not perfect, of course. And when you combine that with a field or a disease where biology is already known, but no drugs are approved, you get a chance to come with a very good target. So the second field where we selected the best technology was also in which disease model are we going to use. Okay, we could have gone for animal models or cell lines, or even primary cells isolated from human tissues. But they all have limitations in terms of productivity or in terms of speed of data generation and feedback loop, or also in the fact that you can control experimental conditions or not. So the best technology for us appeared to be the patient-derived cellular models. So iPSC-derived cellular models, because it's very close to human biology, and you can produce them in a large quantity, so you can generate enough data. You can control the conditions, experimental conditions on that. And a lot of progress have been made to differentiate iPSCs in many different cell types. So we have that in Europe as well. Very interesting, well, I have to say CROs and academic labs that have developed that. So also something that we have here in Europe. And then the third field where we selected the best technology was in terms of drug discovery. So their AI has generated a big revolution from discovering a drug to really designing the drug de novo. And this is the field of generative AI, generative chemistry, that has really enlarged the chemical space significantly, and also now has progressed, because in the beginning there were complaints that okay, AI designed the molecule, but it's not possible to synthesize it. So now the tools out there have evolved towards synthesis-aware de novo molecule design. And the impact is really huge, because you can reduce the iteration cycles, you reduce the number of molecules that you have to synthesize, and you can increase novelty as well. So it's a significant reduction of cost and lead time. So all these technologies together now, in the linear view, have allowed us to create this process, which is our discovery process for first-in-class lead compounds. Starting from developing model cells and producing cells enough to feed into the second step, which is generating the right data, single-cell multi-omic sequencing data, feeding the algorithms and getting the output out of it, which is a list of ranked target candidates. And then for selected target candidates, going into the third step of target validation, so doing gene expression perturbations and functional and mechanistics readouts to target nomination, basically. And then the four steps on the targets that we have nominated go to target to lead. Using structure, prediction, and molecule design. So with this process, we can divide by 3 the time, divide cost by 2, and get to a lead compound with less than 100 molecules designed. So now this is our first PINCO, to make it very concrete. It's the Starlight Therapeutics. So we are it's a SPINCO in collaboration with VIB, with Steinhardt for the in silico part, and in collaboration with the Duve Institute from UC Louvain for the disease biology part with Professor Bommer. And we are reaching now the first milestone 1. So we are still there. What was very positive is that this prediction tool allowed to rank the known targets that are already in clinical development very high. So this is a very good indication that somehow it fits what nature is proving as well. So yeah, that was it for me. So happy to take questions.
Are there questions in the audience? Yes? Start here.
So, always intrigued by this chemical part in things. You're aiming to unlock a space of 10 to the 27. If you want to do that, you need extensive coverage of reactions, which, and the previous presenter already hinted at that, means you need access to a very diverse reaction data set. That sounds like very hard to get by for a young startup. So how do you handle that?
Yeah. So the platform that was built the lean startup way. So not if we would have been developing, our own like GRN inference algorithm, our own CAM-AI algorithm, our own iPSC platform, it would take forever, and we will never got the money to do that and never do anything. So it's more about relying on existing platforms. So I cannot, of course, disclose which platform we are using. But this chemical space calculation is based indeed on the number of reactions, named reactions that are mastered on the platform, and also on the basic compounds that are commercially available. And by combining these two elements, it's not large language model-based platform. This is what I can tell you. So it's not based on that. And this is how they calculated it. So it's their calculation, not ours, and we are not pretending that we can do that ourselves. It's relying on expertise, yeah.
It's not really a scientific question, but more about the cost structure of the 4.5 million for target to lead compound.
Yes.
I was curious to know how you see the biggest weight in that whole process. So what is the most expensive, and without if it's confidential, but it's just out of curiosity for.
Yeah. Yeah, sure. So it depends on the number of shortcuts that you can take, of course. Because if you have, for example, to generate your own iPSC lines, if you have to create your own patient cohorts, and it depends. But there are a lot of shortcuts that you can find out there, hopefully, relying on others' work, on other business models, and so on, that fee for service, and so on. So I would say that in our model, it's approximately 1/3 for the target discovery part and 2/3 for the hit to lead part. Yeah.
Thank you.
Another question? Yes?
Yeah, I was wondering how easy it is to raise money in Belgium. Is the VC space good enough? Is it easy enough?
Yeah.
Are you having to move outside? Maybe France, maybe the UK. I don't know.
That's a good question, and I have to say, yes, it's difficult, and in these times, I think when you come with a concept that makes sense and when you take risks but in a controlled way, so you are proposing to take risks, what we need, because we need to change something in the status quo, the Erum's law in the pharma industry, so people are aware of that. So when you propose to take risks but in a controlled way and with key technologies and key people on board, people are interested. Investors in Belgium are willing to fund initiatives that will position Belgium and keep Belgium really ahead of the field. There is a need to also repopulate the biotech ecosystem, I think, after the last months. So there is a willingness to take risks, I think, when it is a controlled risk and with the right people around. So yeah, we will be fundraising again and again, so each round will be different, I'm sure. But the first round, yeah, we got it quite fast, I have to say.
Thank you. Not that we have a talk from VIB, then Aquila, and we move to an actual startup existing, so it will be the talk of Ségolène now from Quantify. So Ségolène, you're welcome on stage.
Just checking if there is a timer, because I saw we are running late, so should not be punished by Thibaut. Welcome everyone, and thank you so much to the AI for Belgium team to put up this event. It's not a diplomatic sentence I'm saying here. It's really, I found it fantastic to, I see we are not so much, but I see there is a lot of quality, so that's good. And if there is a field where it's good for Belgium to think a bit about how to position ourselves in terms of AI, it's biotechnology and drug discovery, because it's one of the key strengths of the ecosystem, so we should not be let aside by the progresses of others. So I'm Ségolène, the CEO of Quantify, and what we've done over the past few years is build Sapien, is our technology. And let me show you around a bit. So before preparing the talk, I was wondering, but who are going to be the people? Because AI or biotech. So I try to do a bit of a mix, at least to give you some flash, not in terms of scientific or technological approach, but rather in terms of what, thanks to AI, we are actually and practically doing, and the kind of challenges we are tackling for drug discovery. A few years ago, we were already around as an AI consultancy working on different verticals. So our short team, small, was doing work around marketing, around pricing, around a lot of things, logistics. So nothing to do with drug discovery. And then an event, a personal event, happened to one of our co-founders, Nick, who was diagnosed with 12-kilogram cancer, a liposarcoma, which is a rare disease with no available therapy and treatment other than surgery. So we started thinking about what we can do with our brain and our team and our willingness to do things that matter in the field of drug discovery. And this was the start of the journey. A few years later, this is a team, and we have totally switched. By marketing and all these things. Our core business, our core focus, and what we have learned as an AI native company is to do drug discovery thanks to artificial intelligence. So the team started with a couple of AI data scientist engineers, some by training, some self-taught, and then step by step we expanded to colleagues with, of course, a machine learning background, but also chemistry background or biomedical engineering background. Our mission is very strong, is to use AI to improve human health. So our core focus, our core hope, and our core ambition is to really do something that matters in that field. I think around the room, and this has already been mentioned, we can all agree and have a drink at the end of the day to say that even if AI is going very strongly and very powerful, AI drug discovery still remains curbed. So there are several reasons for that, and some of them have been already very well explained, but on our side, one of the key challenges we wanted to tackle, this was the first one, was really to be able to find modulators, small molecules, so we work on small molecules, I forgot to say that, for undruggable targets. So really looking at undrugged indications and targets, we thought, wow, we can go to the moon and we are still not able to do that. Well, let's try to find something that works and that works well and that works at scale. There are several reasons and very practical reasons for current AI approaches to be unable to generalize, and one of them is the unavailability of training data. But so on our side, we thought, okay, structural approaches are here, it's fantastic, but maybe we can design novel approaches that work, and work sometimes better, hopefully. This has been also already shortly mentioned, so very practically again, I told you I wanted I'm too tall. I wanted to focus on I can do like that, or with a mic, like a real singer. Thank you. Thank you. Should I switch up the other one? I will. Fantastic. It's good to be at the end of the AI week, so everyone is very well trained. Thank you. So around the canonical human proteome, only around 5,000 protein targets, therapeutic targets, are currently drugged. So there are a lot of people out there looking at the undruggable and working, including pharma companies, on undruggable targets and trying many approaches, and it's not possible to drug these targets. So our goal is really, with our platform, to find modulators and to drug the whole human proteome. So this is a big challenge. The ambition for our platform is really, so we have we spoke about the data problem. We have spent years and years collecting data about compound protein interactions, about patents, about a lot of data we could access and curate and structure to train a model to really answer to this challenge of whether any molecule will bind or modulate to any protein. This constitutes data sets consisting of hundreds of millions of data points, highly curated. So if it was not curated, it would be much larger, actually. So we started building a first model, which was called Sapien-Hit, and we evaluated it in silico, and we saw that it could have, it was predicted to have extremely good capability for undrugged targets. And then we started opening doors of pharma companies, and they told us, hey, it's fantastic, your thing, but give me wet lab data. So we worked on giving wet lab data. This was totally new ground for us. Remember where we come from. So we opened some doors, and the doors that opened to us were doors of people who, like us, because we come from the rare disease perspective, people who were trying to look and which couldn't look for and which couldn't find any drugs. So we started working with the University of Antwerp and the Fragile X Patient Association, for example. We started working with a few academics and research centers in the field of ion channels or channelopathies. We started working with AFM Téléthon, which is one of the biggest patient organizations active in the field of neuromuscular disorders, and so on and so on. And the middle one, it's here, so you see plants, and you may wonder why the hell is she showing a plant? It's not a patient. No, but one of the researchers, we actually reached out to us, and he told us, hey guys, I'm working on plants. Can you try testing your platform on plants? And we thought, okay, why not? And actually it led to the discovery of first-in-class modulators for therapeutic targets associated with plant drought, so climate hydric stress resistance. But so over and over, collaboration after collaboration, we saw that the platform was having extremely good results, and we were managing to reach this target. So what does it mean very practically? Is that so? If you're lucky with a high throughput screening, so usually one of the wet lab methodologies, techniques used by pharma companies or biotech to discover small molecules is high throughput. So you test physically small molecules on a specific bioassay, and you see if your small molecule is active or not. The hit rate in high throughput screening is 0.2% if you're lucky. No? So it's very expensive, and it's not only very expensive in money, it's expensive in terms of risk, it's expensive in terms of time, and also in terms of environmental impact, because you're spending a lot of reagents for nothing, basically. You know? So of course you can get interesting data because it's negative data, but still it's not effective. So we had evaluated our technology, Sapien, already in silico, but over and over, when we evaluated in different compounds, in different therapeutic targets, sorry, we see a performance from 4% to 96%. Huh? So what does it mean is that we usually work with our academic partners. So this was the beginning. Now we work with more with partners who have better capabilities, but sometimes they told us, okay, I can test your compound, but you can only send 10 compounds. 10 compounds. What can you do with 10 compounds? Nothing. It's like you're just bound to fail. But with 10 compounds, for many projects, we still had. I have another example later. Some projects which succeeded. So 4%. It was yesterday. We got it for an ion channel. So it was actually our lowest score, but I thought, okay, I'm giving this presentation, I have to be very transparent, so I decreased, it was 10%, and now it's 4%, but it's important to be reliable, right? And 96%, it looks very unbelievable. I will show you later what it corresponds to. It's for an already liganded target, so we had a bit of training data, of course, but still 96%. And it's still very surprising. So the technology, based on totally different concepts and different approaches, but a lot of data, manages to have this impact, which is completely, which is remarkable. Huh? So now let's take again. So I mentioned this high throughput screening that I guess many of you know about. But high throughput screening is limited. High throughput screening, if you're equipped biotech, you can do, I don't know, 100,000 screening, maybe more, depending on your resources. A pharma high throughput screening campaign will test around, I don't know, 1 million compounds. So when you look at, if you translate a very difficult translation, compounds into rice pieces, into rice grains, you will get a high throughput screening campaign represents, if you take 100,000 compounds, a bucket of rice grains. What we try to do is to use Sapien, our technology, to screen, of course, in silico, higher libraries. So when we started, we used to screen MQL, which is a library consisting of 8 million compounds. And we already got good results. And we thought it was time to switch to not the small bucket, but the big lorries. We switched to enamine reel last summer. And so we can screen in 4 hours 10 billion compounds. And we keep the same kind of hit rates. So in 10 billion compounds, we are able to find a series of totally novel and diverse chemotypes that modulate our target of interest. So the technology is quite useful. Then I have little time, but Florence mentioned this problem of herding, and also sometimes it's very difficult for when you are looking at already liganded targets to go very far away in terms of chemical diversity. And here, but I don't have time, but I'm showing you on a Tanimoto heat map in terms of chemical distance, and you can see here, so it's for, I don't have time, but for each compound that we find, we have extremely different chemotype. So it's a novel basis in terms of chemical diversity. What does it enable us to do? Remember, our mission is use AI to improve human health. So it was last autumn, I think, we announced extremely good in vivo results in liposarcoma, so the cancer of our colleague Nick. Where we actually, in that case, looked not at novel molecular entities, novel compounds, but repurposed drugs, because we are patient-driven. So we want to find something that is quickly available. The drugs have nothing to do with cancer. We identified novel targets, but I'm not going to go into the field of target discovery because it would be too long, and I'm already very much above time. Another case is a rare autism spectrum disorder. Super tricky target, no non-modulator, no non-3D structure. Round 1, 10 new molecular entities tested, a case for failure, but actually we had 3 weakly active compounds. And then round 2, optimization, 2 lead-like candidates with IC50 between 2 and 5 macromolar. So really something that a platform that also enables us to go very far in terms of lead optimization with very low resources, which in the field of rare diseases, but also targets that are not very well known and for which assays are not available at high throughput, it's extremely positive. Last one is NAV1.8. NAV1.8 is a very well-known well-known target in pain, non-opioid pain management, and we have 4 novel chemotypes. So a lot of cases. We have 16 projects on our website. You will see, and a lot more going on. And on that, I think I'm almost on time, and this is our team in full shape. That's it. Thank you so much.
The questions online? No? Maybe I'll start by one question. Can you explain a bit what is the business model that you develop here? Do you intend to develop products, or do you?
We do both. Huh?
Yeah.
So we started last year some collaboration with pharma companies where we co-discover small molecules. We focus really on the small molecule discovery and amine screening. I didn't speak about that, but we also have a couple of models for ADMET-tox discovery, for selectivity prediction, so it goes much further than just finding novel chemotypes. Also patent exclusion to be really IP-free. But on the other side, we also have our curated small, because we are still a small company, number of assets that we develop step by step, but really using our platform to advance in some undrugged targets or targets for which we know that there is a high interest in pharma, so we can use that to grow further. But so we do both, and we think it's good to be the users of the technology because we understand really also the boundaries and the issues. So it's in a product development mindset is very good.
And you intend to keep both models?
Why not? This is an interesting topic, actually, the field of tech bio, so tech bio, biotech, so tech-powered biotechnology is, the business models are, we have the luxury of being able to look at what others have done and where they have failed or where they are succeeding. Our model is right now totally different in the sense that we haven't raised, unlike the big. I saw the big news as well. for Xaira, but we haven't raised any funds right now. We are totally bootstrapped from toe to head. We are really different in terms of model as such. But yes, the duality between product development and being AI-powered biotechnology, we see it company after company. We'll see. We'll learn. We'll still learn from the others in terms of what is the best model to do.
Thank you. Another question? Yeah. Yeah.
So you said you're looking for new implementation for AI.
So, once again.
Hello? Okay.
Yeah.
You said you're looking for a new way to implement AI in your business, or you already have people who work with you with AI tools?
Yes, we have a team of AI machine learning engineers in the company.
Are you looking for something else? New ways? New
right now,
implementations?
but it's another good question. No, not yet.
Not
but
yet, okay.
we receive with pleasure applications. But so right now, our strategy and our approach is not necessarily to grow in terms of. It's a different kind of answer, but in terms of people, but rather grow in terms of technology capability.
Yes.
So a platform that can scale towards across targets in real time. So in conclusion, as a consequence, we are not recruiting right now, but we know that we will recruit at some point. So.
But you are open for somebody
Yes.
who has new tools to show you how to implement AI in your business, even though you don't.
If it's
hire
drug
it.
discovery, yes, or something else that can fit with all whatever. What I gave you a very high level
Okay.
view, but
Thank you, Ms.
it's
Congratulations.
good to-
Thank you.
Thank you so much.
Well, let's applaud the speaker once more. Thank you, Sigurden. And now we will welcome Hugo Clemens from J&J. So we will hear a different perspective on how a much larger company intends to innovate based on AI-driven drug discovery. Thank you, Hugo.
Thank you. Yes, just it sounds like it's not on yet. All right. Yes, now it works. So everyone's still awake. Friday afternoon, sun is shining, so I have some competition. It's good. Who knows J&J? Who does not know J&J? So, really big healthcare player, tech dev on the one hand, US headquartered, but the other division is innovative medicine. And we should be proud in Belgium that a Belgian company, Janssen Pharmaceutica, was at the very origin. So it was the first company that started pharmaceutics. We have already heard it from other speakers. Drug discovery is hard. It takes 2.6 billion to take a drug to the market. Lots of numbers there. And can AI ML help with that? The answer is yes. And we need technology on the one hand, and we need implementation in the cultural sense, embracement of those technologies to put it there. So in the first part, I will focus on that implementation. So where did we really bring AI ML, not just as a technology, but really put it into the hands of chemists and biologists? In the last part, I go a bit towards the technologies we are looking forward to that are still a bit more experimental. Most of the examples I will show are small molecule, like everyone else in here. But I want to stress we are doing similar activities in pretty much every therapeutic modality. So that means not just small molecules. That also means peptides. That means degraders. That means antibodies. That means RNAi. That means gene therapy. So everywhere we go a similar path. And I won't have the time to go into that, but if people have a few questions on that, I'm happy to answer them afterwards. So, where did we start? So small molecule. Up till, well, in the year 2000, we were like everyone else. Yes, we had a really good CAD department, chemoinformatics, QSAR models, structure-based drug design. We did start dabbling with AI/ML in 2013, but it was much in this pilot way. Okay, this is this new thing. It's interesting. Let's have a sandbox experiment. And then actually, by COVID was a bit of a help because people had some time to, well, we're at home, so what are we going to do? To actually say, let's bring those methodologies not in a sandbox, but really in concrete projects, using different property models, image-based predictions, which was new at the time. By 2025, we now are using generative AI in small molecules, in proteins, we're going multimodal. And by 2030, and it already hints towards the end of the talk, we think strongly that one of the things that's going to happen is that AI/ML will unlock more automation. But let's start with what is already in place today. So 3 very concrete examples. The first one is a thing we call Sentinel. And Sentinel is about getting to know the things you don't know. So everyone knows that they're looking for the effect on the target you intend. But how does a drug company come across early safety signals? That has very often been a challenge. And if you wait until the first compound goes into an animal, you're very often late. So one of the things we have completely implemented, every single molecule before it is made is screened through models. So you know up front whether the machines already have a hint at, okay, looks safe, looks unsafe, or for the love of God, I wouldn't be able to tell. When you make it, every single one is then assessed. So based on structure first, should we actually test it? Because we don't know, and we want to learn, we want to pick them up. Then in a second route, we put every single new molecule into a workhorse assay. We get a microscopy image, and we do the same thing again, but now on models that are trained on images. So if you don't know the structure, that's not so bad. If you recognize a phenotype, you could still capture that. These are very concretely in. Every compound that any project makes, even if they're completely unaware that there could be a risk, can get flagged either based on phenotype or on the structure itself. And that is automatically built in. So if you're flagged, then you can actually raise that. And this new thing, this new criteria becomes part of our design criteria. That's completely embedded. So everyone who makes molecules themselves, so every synthetics is subjected to that. The second part is lead optimization. That's probably the oldest part where we already had activities. So this is a slide that actually was not made by me, but by one of my CAD colleagues. So that's a department that for a long time thought, well, we have been doing this for decades. So if they start to introduce some of your methods into their own slides, you know that it's being adjusted. In silico design strategies are now formulated for every single project. There are very traditional, right, like reaction template-based enumerations, but then there's this little box machine-assisted enumeration, and that goes into that bucket of synthetic chemistry. So rather than like in the previous generation, first getting interesting compounds and designs out, we start front-loading that upfront. We use CAD technologies, molecular descriptors, docking, unwanted groups, etc. We also use in the middle, you see there a bucket. This is really a slide that I just took from a presentation. That is everything that enables design, human dose prediction is used during this project, and it turns the compound at the left-hand side to something that is, one, more drug-like and also more potent. And then we combine it with more traditional approaches like conformational modeling, chemical similarity, to deal with selectivity, because that was the thing that AI/ML was not good at nailing down. So this is a very concrete example. It's a stopped. That's why I was allowed to use the slide. But it shows you it is being used in practice. The last thing is human dose prediction. So this is about, you know that the compound works. We already start having an idea, should it have issues or not, but what is now going to be the dose? Traditionally, you take some ADME into account very early, but then it's waiting until you're fairly late and you start putting compounds into animals. To then get an idea, is this being well absorbed, and what should I do if in the future I go into humans? So we have an entire network that allows us from the very first time you make the compound, even if it doesn't exist. So you only have the designs, you can start ranking and using a number of assumptions purely based on that theoretical structure, you already start doing rankings. We have lots of data in ADMET, and then as you go down the funnel all the way to animals, it becomes less and less and less. So there's there's transformer distillation networks that take care of that. Then there's an AI version of single species allometry to then project that. At the right-hand side, that starts illustrating as we now get more data. So the people have made their first attempt. They start ranking things, they start making compounds, they get a bit of experience, and we collect more and more data. All of these models can be updated with the real data, and they adjust. What you see on the graph there is at the y-axis, it is the prediction if you have no structure at all. So this is at the very first day. You think I'm doodling up a structure. What is this going to do? If I put it to a rat, what's going to be the dose there? On the x-axis, you see if you have a lot of readouts already available. And you see the correlation is not perfect, but it is already helpful. So from the very first day, you start already driving towards getting decent molecules in. So these are things that are today in the hands of every project, every small molecule project, and we're working to also extend that to these new modalities. This is a much more recent project. I cannot disclose the target. This is PROTAC. PROTACs are really big molecules because they have to do 3 things at the same time, so it's 2.5 times bigger. And as a result, because they're big, it takes a long time to make them, so your design cycles are long. And secondly, they don't enter your body very well, so they're harder molecules. So this is an example where we had a massive molecule. So this is, if people are familiar with the rule of 5, that ends like, well, anything bigger than 400, 500 becomes being a bit hairy. Think 3 times that. 9 million in numeration, it goes down to 140 compounds. We sent 39 to synthesis. Based on all of the modeling work, 9 were put into the animals straight, so no further ADMET testing, just straight into the animals. 8 of them had double-digit oral availability. 1 of them had the biggest oral availability that was ever recorded for that particular E3. This is complete in silico exercise. You don't do a lot of testing. You go really fast and then go to the animal really fast. The numbers are lower than you would have in the traditional approach when applied to this class. So this is really helpful. I'm now shifting to the things that are that we are exploring, but that haven't been put in the hands of every project team yet. We are piloting those and validating those. And this is the line along which we do that. So there's always a team that is prototyping the new tech, then they start piloting it in a handful of selected projects, then they start assessing based on the results, does this have value for our business? Is it working? And sometimes it does not, and then we kill it. But if it's working, we productize the tool, and only then we really push towards integration like I discussed before. One of the things, and we hesitated quite a long time, there was a discussion on foundation models, earlier language models. Language models don't work really well with chemistry. They really don't. Now, if you extend them to work with graphs, or with tensors, etc. So it is still transformer technology, but you push it a bit further. You can get further, and you can really start using all of those compounds. I mentioned before, every compound that's made, we throw into an image. That's a lot of images. We have the classical descriptor. We also have quantum computations for them, and we use those. So we feed all of that in this large graph former model. That takes quite a bit of compute, and these models literally train for months and months and months with checkpoints, etc. The nice thing is, just like ChatGPT, you can prompt them and refine them on the fly. So when you use them in a project, you don't have to redevelop them from scratch because they've seen every single data point you have. They have seen every image you have. They even have seen reactions. They have seen QC. They have seen all of those things, and we constantly keep monitoring how well does this do and how well does this adjust when we stop. For some of the properties, you only do mildly better than a classical, and this was mentioned, than a classical linear. So why are we still interested? This is one of the examples. So on the left-hand side, you have log D. On the right-hand side, you have EPSA. We stop these things training at a certain point. If you do that with a linear model, it goes down. And we were surprised here, okay, first quarter, this still seems to be doing good. So we stopped training this thing in 2022. Now, our production model is still trained, and it gains still in performance, but we keep the model that is not being trained, and we keep seeing. And you can see over time, this is holding up pretty well. So no training since 2022 and up till 2025 here, but if you would continue that up to now, it still performs the same. So one of the advantages of these massive models is they become way more generalizable. If we now go to new modalities, so we have an awful lot of data on small molecules. But what if we go to those new modalities like peptides? Over here, the model is trained on small molecule data only, and we just start throwing peptides in. So that's different from going with a molecule of 80 to a molecule with 500. Out of the box, we see something that is predictive. And when we then start adding those peptide data, it's like adding a tiny shot glass in a massive wine barrel. You see that suddenly, so both, we did expect that the peptides would be a bit, but what we did not expect is that adding the peptides actually also improved the small molecules. These are the things that start happening if you build these big models. There's a lot of cross between the compounds. I'm going to wrap up now with the last two things. I think the questions I asked already indicate I am interested in where is synthesis going. This is not by popularity vote. Chemists are always a bit wary because synthesis is their thing. But if we want to go towards the future, automation, particularly of easy reactions, is going to be key. And if you want to automate, your robot has to know how to execute reactions. So one of the things, a Suzuki Miura, for people who are not interested in chem, it's a coupling that is very frequent, but typically after doing some experimentation, so you do a small experiment to explore a few conditions on one representative, you can get 80% success rate. If we do that with machines, you don't have to do that first experiment, so you save a day, and your success rate goes up from 80 to 95%, something like that. Now, what if you do that with a Buchwald-Hardwig? That's much harder, because there you don't find a single unified condition. So now you would have to do your experimental optimization on each individual well, which becomes prohibitive. You just do that in a computer, and then you send that for automation to execute. Where a chemist typically would have 25% success, and they would have experimentation up front. Without experimentation, we go to 65 straight. And then the next step is then automation. So I think I'm reaching time, so I'm going to wrap up here. Still believe that there's a lot of opportunity here, and that's needed because so much more to be done. The patients are waiting out there. Happy to take a few questions.
Thank you, Hugo. We have a question there.
So I heard, first of all, congratulations for your presentation. I heard you spoke about risk, safety risk, when you started your presentation. But there is a new way now with AI automation, we can fix those risks by doing risk assessment, AI workflow risk assessment. So if you're interested, I can show you those ways that will reduce your risk. You don't have to fix the risk, but you prevent risk. Before it happens, already fix it. So I'm here to tell you, if you are willing, I would love to show that new ways, new AI workflows that help us to reduce risk by doing risk assessment now. That's what's going on. So you don't have to wonder about how you're going to fix the risk. You prevent risk. No matter how the risk goes, it can go higher, level, small. You delete to those risks.
It sounds like a great topic, but I think it would run out of the time span we have here to answer questions.
Yeah, I know. I'm just
so I'm happy to afterwards over the drink spend a few words on that.
Thank you, sir.
Thank you. Another question? Yeah, two over there.
I had a question about the next frontier, because you have like horizontal view because of all the therapeutic modalities that you're developing and discovering, about the use of AI in drug discovery. And for you, what are the toughest challenges? I know it's a very wide question, so you can just pick whatever comes to your mind, but in terms of AI applied to drug discovery.
Yes. I think one of them is automation, not just in synthesis, but in a lot of tasks. And that's not automation, just the machine will do it all. But we have so people do still too much routine stuff. So we have to free up people to focus on novelty and have a lot more things done. It's like a combine harvester. In the past, everyone tilled their own soil. Nowadays, if you want to stay in business and have a combine harvester, so automation is the first thing. That is more complex than people give notice. The second thing, and I mentioned that on that small slide, we do believe that we do need other targets. We do believe that most of the targets are accessible. They're not accessible with a single modality. So can we extend all of these tool sets to all of the modalities? And do we have a good idea when to use which modality? So those are two questions.
Thank you.
Thank you. There was a question there first, Eric.
Very nice talk. In the beginning, you said that for antibodies, you were going towards multimodal models. Is there anything more you can tell us about that?
The multimodal actually was broader. It's not just on antibodies, but so multimodal here for us means multimodal in the sense of multi-therapeutic as well as multi-data input. I hinted at a bit of that if you start describing molecules, any molecules, with either structure or sequence, or if you start describing it as a data-rich readout like a microscopy image. But you use that as an input, not as a label. That's a second modality, and obviously you could think of a few phenotypes that are amenable. Now, obviously, size comes. So there's a lot of nuance to that answer. But yes, we're looking at both and trying to leverage them as well as possible, and that gives us some results.
Thank you.
Yes? A lot of questions. Yeah. Here you are.
Thank you for a very interesting talk. So these generative models are statistical models, right? They produce data depending on the probabilities that are representing inside the model. I know you're a very big expert in statistics. I remember you from IWT panels we were in together a long time ago.
Yes.
So can you elaborate a bit about the biases that you see in these kind of models? Because, for instance, if you look at classical text-based language models, and you would compare them, for instance, to humans, how they decide on certain things, you see they are extremely skewed and biased to stereotypes, what they've seen the most. I mean, there's a lot of overfitting going on in those things. So could you maybe elaborate a bit on that, on how you counteract that in your systems, if you can?
So one of the answers is the multimodality. So they're also very biased, but you're starting overlapping the different biases, and that's very useful. So where you see chemotype bias in many of the structure-based approaches, you don't see them in the pheno-based approaches. The second thing is bias is not always bad, because we heard a few people mentioning enamine. Enamine becomes uncontrollable. So the enamine grows faster than the methods to process them are evolving. So where we do see the future is more like an enamine on the fly. So you will develop your own virtual libraries that then are synthesis aware, so that you can execute them on machines. So we can't handle 10 to the 60. We can't handle 10 to the 20. But so it's more going in towards those pockets, and those biases actually can help. So it's more like you go in, and then you have a massive explosion in a small area, and then leverage those, and having a few orthogonal angles by combining things that are trained on different input sets. That's at least where we're probing now. Future will tell whether that's the right gamble.
Thank you. One last question.
Yes. So thanks for your talk. Two questions. First, can you share what is the size of the chemical space you can handle at J&J? And second, is there an estimation that you can share of the adoption of AI in your projects? Are you using AI now in each and every project?
So to the last question, in certain forms, yes. So there is no project that escapes AI anymore. That I can be adamant about that. The level of interaction is different. So we still pilot the latest technologies on a limited amount. And there we go full throttle. So there's pilots projects that get more support and where we throw everything in the kitchen sink at, so to speak. Does that answer at least the second question? Yeah, to the first question, so in principle, you can do everything. In practice, it is limited to which reactions can you handle, but also what is the applicability domain within each of those reactions you can do, and what's the transfer. And there, having alternate descriptors that can help you helps a lot. We still are very data dependent, but there is the decision where we feel the biggest need is. So the most frequently used reactions where we have lower success rates and where we do see that models start picking up, we invest a lot to get those prioritized. And we have plans to extend that gradually. But already today, about 50% of the most frequently used assays, we would have a useful model. I'm not saying a great model. I'm saying a useful model that already statistically would give you the leg up.
Thanks.
Thank you again. So we have heard that no project can escape AI. That's very, very good news. So now the last speaker for today, Romain Alderweireld from the Fondation 101 Génome, a different angle on the question of AI in drug discovery from the standpoint of a research foundation. So Romain, welcome on stage.
So you see the slide? No? I can't use this one? And this way? Not yet? It's coming? Yes? So, as we just heard, the patients are waiting. So here, I'm representing the patient. I'm Romain Alderweireld. I'm actually a lawyer, so I'm really honored to be here with you today and to end this week on AI, because I don't have your capacity and your knowledge. With my wife, Ludivine Verbogen, we created the 101 Genome Foundation. And I will put a bit of context to understand why we use AI since several years now to help and to accelerate the availability of new drugs. So everything happened with the birth of my third child, Aurélien, who was born with a rare disease. And not only with a rare disease, but a really severe form of a rare disease, with a life expectancy in 16 months. He was 11 months when we heard about that, and it was a terrible shock. Hopefully, we had the help of somebody called Professor Guillaume Smits from Erasmus, and he was really dedicated, and he took a lot of time to explain us and to learn, to Ludivine and I, a lot of things, because we knew nothing about biology, we knew nothing about genetics, we knew nothing about the genomes and the 20,000 genes in the genomes that interact between each other. I was unable to sleep, and I decided to study science. And with the help of Guillaume, I dig into science and I decide to conduct my own experience. Doing this experience, I cross two databases. One database with people, what we call control people, harboring variants that are not causative for disease. The story of the cat and the crack that Florence showed you earlier. And on the other side, I use another database where we, a group of people, gather pathogenic variant on the gene FBN1, which is the gene which is affected by the disease of my son. And at the intersection of two groups, I identify a group of 24 patients harboring the same kind of pathogenic variant as my son. So those people should be sick, should die really early in their life, and they were apparently quite reasonably healthy adults. So I went back to Guillaume, and I said, Guillaume, what is this story? What is it about? And he told me about the impact of the modifier gene, the story that we talk about, the network of genes, the interaction between the genome within the genome, which is sometimes able to counteract even the more deleterious effect of a pathogenic variant. This discovery I did years later, it was confirmed in another publication, so we can go in this way. So with this, we understood that somewhere in the genomes, there are other genes that are able to counteract even the more deleterious effect of the disease of my son. So my wife and I, we wanted to go back to the data, but for legal reasons, it was impossible to go back to the data of the control. So we decided to create our own cohort of patients, try to identify ourselves those pathogenic variants and the modifier protective genes. So we created the 101 Genome Foundation with the support of the King Baudouin Foundation. We gathered and we created our own scientific committee with some people you can see today in the room, and with them we decided to elaborate a research project called GEMS. And the main idea was to gather a cohort of patients harboring the same disease of my son, with some people being quite old, with few impact of the disease, and on the other hand of the spectrum, people being really affected really early, like my son. So we decided to go for two directions. One, we called it the wet lab, traditional iPSC discussion that we heard today, and the other hand, we wanted to leverage on AI. This is something that we created in 2016. So with the 101 Genome Foundation, the first thing that we created, it was we opened our own biobank. This is a place where we gather the genomic data, the biological data of the participant to the research. On the top of it, with a researcher from the MIT, we created the first of its kind genomic cloud in Europe, where we gathered the genomic data of the participant to the research. The genome, what is it? Is it the digitalization of the biological information in the DNA? So we turn the biological DNA into a digital DNA that we put on our genomic cloud. Being a lawyer, my wife and I, we were really, really unsatisfied with the way the consent is gathered for the people joining the research. And we see that it was a real impairment to the research in the field of the rare disease. So we create our own solution, which is an app that is really available and used now worldwide since more than 3 years, that allows patients with rare disease from all over the world to join directly and to interact directly with the main investigators in the field of the disease. The system is complemented by a system where we can collect, gather the biological samples from the participant, and so from there we can extract their DNA and sequence the DNA. We do this thing in collaboration with the UZ Ghent, where we have the extraction of the DNA, and when the DNA is extracted, one part goes to our biological biobank, and the other part goes for sequencing in our genomic cloud. What's really important in this system is the consent and the fact that the participants are really involved in the research. So we have a bidirectional portal, which is called MIDA, and there the participant, they can always find back their consent, withdraw their consent, extend the consent, provide additional data, and it's really a dynamic consent, which is actually the solution in the field of the genome that is promoted by the World Health Organization. So we are not a regular patient association doing just awareness. We are really in between the patient association and the different group of research. And here you have the different patient association that join and support our research, and on the other side you have all the different scientific teams that are working on the data that you are able to provide. So here we are really in between. Earlier today I heard about fair data and participation and sharing. If you want to do that, you need to respect the Data Governance Act that needs you to be what's called a data altruism organization. We, the 101 Genome Foundation in Belgium, we are the first data altruism organization recognized at the European level. Forever we'll be the first, okay? So we respect this system. So where we are today with the 101 Genome Foundation project, we have more than 230 participants who joined and shared with us their genome. We receive here in Belgium genome coming from Belgium, France, Luxembourg, the Netherlands, Slovakia, Sweden, Norway, Austria, Spain, the UK, Denmark, Switzerland, the USA, Australia, Mexico, New Zealand, Malaysia. And today I understand that somebody from China will join. So when you say that the fact that here in Belgium there's an advantage with the data, this is quite true, because now the US foundation, the American foundation, they rely on us to gather the genomic data from the patient with the disease of my son, which is the Marfan syndrome disease. We have a complete process that's really effective. Sometimes it goes really fast, sometimes it's quite slow. It depends from the participant. We don't want them to press them. We want them to take their time and to join when they are ready. And this is one of the strengths of our system. So as of today, we have 234 genomic data from participants. We received 111 exome data from INSERM, and we received more than 638 panels from all different groups from all over the world, meaning that we gather the data from almost 1,000 participants in our cohort, which made it a wonderful place to use AI tools. So, the first fact was the birth of my son Aurélien, the discovery of the disease. The fact that you understood that the patient organization, they have more to do than just wait that the pharma company will save them. So we created the 101 Genome Foundation with the support of the King Baudouin Foundation. We opened our biobank. We create our first genomic cloud solution. We create this solution to gather the data with this the GEMS app to manage the consent, which is the real hurdle to the research. And with that, we went back to the patient association and feed the complete loop. We understood immediately that with the genome, we have a clean data, a digital data. It's not sufficient, it's not by itself, it's not alone. It's a third path. It's the in silico path that we want to add to the in vitro and to the in vivo path. And so we wanted to use it, and as first, the first thing that we did, it was this story of the causality, the cat with the crack. We wanted to be sure that the pathogenic variant that we identify on the FBN1 gene are really pathogenic. And a bit like the cell-tune solution that was presented by the VIB earlier today, we create a specific tool that allows to predict the pathogenicity of each and every possible variant in the gene FBN1. So it's a bit like having a padlock and try each and every solution with AI tool and generate and predict the effect that this mutation will have on the functionality of the gene and this ability to convey the message it was supposed to convey. Okay, this is where we were. We created this tool. It's fully available. It's freely available online. You have the link here. It's currently used by several labs that are doing diagnosis. It's helping them to pose a diagnosis in the field of Marfan. We thought it was a wonderful idea, and we were not the only one, because during the same time, Google created something called Alpha Missense, who's doing the same thing, but not only for one gene, but for the complete genome, for the complete 20,000 genes. Still, our solution, in some specific case, can improve the result for Alpha Missense, so it makes a lot of sense to use both. So it's why on our solution you can also find our prediction and the prediction from Alpha Missense. We decided to add to this one the improvement from AlphaFold that we discussed a bit earlier. And we realized, as it was pointed by several people during the discussion today, the limit of AlphaFold and the fact that we are in a world which is really biased and we need to go back to the crystal. And when we look back to the crystal, we really see things in the world of FBN1 that shows that there's a real limit using AlphaFold. Still, in its biased universe, we can see this coherence there, but we understood how important it is to understand really the strength and the weakness of the prediction. What we wanted to do is to identify the different gene, the modifier gene, interacting with the FBN1 gene. So when we have the monogenic tool, we needed to have this oligogenic tool. And there in Belgium, we are really lucky to have Professor Tom Lennart and Nassim Versbragen, who are working on incredible and really unique tools that allow to create the link between the genome, between the different variants, between the different genes. And we implemented with them their tools immediately on our genomic load, which allow us to try to quest and to identify the modifier within the gene of the different participants in our research. Here, we are really proud to show you this one. We will not say too much about it, but in a predictive world, we feel some hints that perhaps we could have identified thanks to those AI tools, possible modifier tools. So now what we have to do is to identify potential therapy, potential new drugs. And the idea is to work on drug repurposing. To do that, like every time, we decide to recreate our solution, and we create a huge tool which is called Nafacola, that you can try to use if you want, where we gather all the information about all the drugs that are available with Shamble, and we try to identify the different market definition of the drugs, the off-label potential utilization of the different drugs, and if there was a room for drug repurposing, which is the main key that are of interest for us. So on the side of the market definition, you can explore the tool. You will see that it allows you to explore it by commercial name, by the name of the substance, and try to define a market. And in each one, you can analyze it. And with it, we used AI tool to generate summary of the functionment of the different drugs. Okay, this is functional, and you can use it. And from there, we link it with the different events that happened in the patent world and the different events that happened in the legal world with the different positions from the European Commission and the Court of Justice. On the other side, exploring the possibility of the off-label use, we use the targets that are used by the drugs and see if they could use in the context of other drugs that really help us in the identification of what we would want to use. And as of, this is the last one, this is the one on, we are looking for Marfan syndrome, we are looking to these different mechanisms that we identify, and we will try to create a new drug. On the top of it, we realize that often the drugs, they have to show and to prove the way of working, and so we are using another AI tool, which is called AORAM, that helps, this is done with Loris, that helps to objectify the measurement of the aorta, which is really a challenge in itself and not something easy. So, thanks to AI, we hope that we will perhaps cure my son and the others. And so we really wanted to add this third path. This is something that we want to accelerate. We don't just want to wait. We want to have a solution as fast as possible. So we also have access to iPSCs and to mice and fish model. Everything we did it in a way that we can use it again. It's not dedicated only to the disease of my son. It's not only dedicated to Marfan syndrome. We can use it for other rare disease. So we have 4 pillars, and they can be used and reused again and again. This is our team with Ludivine, Thibaut, Gabriel, and Eneko, who are here today. If you want to speak with them, they are there. So our next target is to reach even more genome and enroll other rare disease in our journey. And thank you.
Thank you, Romain. Very impressive.
Thank you.
And there is a question in the back.
It's very interesting. Yeah, I have actually many questions, but I will only ask one or two. First, congratulations for being the first to have this data altruist organization. So in the European health data space, there is a big part of the text about this, but nobody really starting it.
Yeah.
My question, short question is why? Second question is, so this is great what you are doing, but the problem, one of the main problems is the fact that consent is so complicated to obtain. And one of the sub-problems is that people can always change mind. They can decide to withdraw. Is that a potential problem for you or not? And the last question is, now we have kind of sophisticated rules for consent, but in the past, practically there was no consent, and you have data in big companies, especially by IQVIA, billions of data. So did you think already about trying to push this kind of company or this company to share their data?
Okay. Regarding the first one, why nobody else is doing it, I believe because they are really just afraid of data and data sharing, and a lot of people believe that they will be able to monetize the data and they will generate money on it, and I think it's a huge mistake, but it's what they trust, and it's why they don't want to share. But we want to share. This is not our data, they are the data from patients, and they trust us because they wanted that Tom work on those data, they wanted that Bart work on those data, and not us. So we are really a real data altruism organization, and we really believe that it was a missing piece. As soon as the GDPR was created, this concept was missing. Okay, so it came as a corrective as a corrective, and we hope that other people will understand the value of it. I heard VIB saying that yes, we are really fair, we are really perfect for verification. Have a look to the Data Governance Act in the context of the question of Mr. So this is the first one. The second point regarding the consent. The consent is crucial. A lot of people in the discussion that we even had here today, here in this building, they say, yeah, but there is a consent fatigue. This is annoying the people to consent. I told them, yes. And there's also the voting fatigue. If you don't want to ask people to vote, that will be really, really easier also. No, the consent is key, and we need to have the consent. And we need to have tools as the tool that we share with you today that allow people to withdraw their consent. This is, we experimented in my life with my wife. The first time we enrolled Aurélien in a study, we received a piece of paper and we lost it as soon as we signed it. And it was really unethical and unpractical, and it works really bad. And third question regarding IQVIA model, I don't know, and I will not answer to their model.
Another question? Yes.
Excellent talk as always, Romain. Thank you very much. Now, so maybe you should stress also one thing, that now that you have this consent tool, which I think is like a huge breakthrough, and everybody should start using it. You know my opinion about that. You discovered another problem, right? Because for Loris, we want to go now to the data to the patients, and the patients say yes, but you have another bottleneck now. Can we maybe elaborate on this problem that people are aware of this?
Yeah, it's linked with the first answer to the first question. It's that a lot of hospitals, they see it, and we understand it, and it's really clear. When the patient trusts their doctors with their data, they want the doctor to be, how to say that, responsible with the way they collect the data. But on the other side, the doctors, they work with hospitals. And you have some hospitals, they really foreseen, perhaps people like you, they hope that they will be able to generate some funding from the hospital, selling the data to different pharma companies, and they are not willing at all to share the data, even with the consent from the participant, which is quite crazy. So we spend a lot of time to go back to the different legal group within the different European hospitals and share them with them the consent from the participant, explain them that we have the consent from the participant, and explain them that they have to provide us the data that we are asking with the authorization of the participant. And it takes a lot of time indeed.
I think it's important to And just because people are not aware, even if you have the consent, there is still this bottleneck which blocks open research for this kind of thing. So,
Is there another question? Yes.
I have a question about the next step. So you have identified a couple of targets, or one target, right? And so next step is to develop binding assays or phenotypic assays. Have you already clarified those?
During your presentation, we were discussing with Gabriel about going back to you about that. Yes, so we have different paths that you are exploring right now, but we're really willing to speak about more that with you, Ségolène.
Yeah, sure.
Thank you. Anna?
Yeah, I was just wondering, I saw these DNA genotech boxes on the pictures. Are you selling the sequencing to patients that are willing to have it? And if so, are they are they willing to pay the full price to get a whole genome sequence, 60, 90 depth? I don't know.
I see your question coming from your previous life, Alain. But no, we don't. Actually, we don't. We have some people ask for that. Some people have been asking for more than what they just received. And this is something that we, at this stage, we say no. And it's only through the doctors and the main investigator. In this case, it's Professor Bart Lewis. And so if there's a specific question about that, they can go back to him. The idea is that if we do an incidental discovery, we go back to, if, for example, when Tom is working on the data, he discovers some things, we inform Bart, and Bart, we have direct contact with the participant, but we require him to provide us the contact of his main doctor. Okay, so if we identify something, the first person who will be contacted is his main doctor that will decide if it's relevant for the health of the participant, and at the end we share it with him. But yeah, let's talk about that. Perhaps you have some great idea and that we could implement. Thank you.
No more question? Thank you again, Romain. And so we are almost at the end of this very interesting session. So I will invite Mr. Ramakers, chair of the FPS Health, to conclude the session for us.
Don't worry.
Okay, Friday afternoon, session is running out, sun is shining. I did send my avatar at 2 p.m., but as happens regularly, he starts hallucinating and he got lost. We're still looking for him. No, not kidding. I really enjoyed his presentations, to be honest. It took me back 25, 30 years ago when I was also still in the research environment, long time ago. And at a certain time point in my life, I went into healthcare policy, government, politics, and in the meanwhile, EU, WHO, and so on. So I will be a little bit touching your field, your line of work, but also emphasizing the larger world we're living in. Some things have already been stressed, EHDS, data, a lot of things to do about GDPR, and a lot of discussions we're having. So for me, it's an honor to be here, and especially to, again, have a confirmation on the wonderful country we're living in, the ecosystem we're living in. A lot of brains we do have in our country. So we did see research, spin-offs, farm industry, which we're quite proud of in our country. and also very specific, very special. I was really surprised, astonished to see that kind of initiative, but this is also Belgium. So we should be proud of our ecosystem and also the collaboration where we have been witnessing also today. Now, AI for health is also related to something maybe new, also a little bit based on the geopolitical situation or the new times we're living in, also on what we call the EU sovereignty. Increasingly important. I think in healthcare, AI is really a disruptive technology. We're clearly not prepared for what is happening already. It will have benefits, but clearly also risks at least level of patient safety and some other issues. I think that we saw today that for drug discovery, indeed, it is a promise, but a lot of hurdles still to be taken. It's also, as we see in healthcare, it's maybe also a little bit of hype. Can you imagine drug discovery without AI? Probably you can, but you will not put it on your website or your portfolio, I guess. But again, I think, especially in Belgium, we should be keen to that innovation. We should be really ambitious to be the front runner in this development, because again, we do have that ecosystem and we can put everything together. Now, Europe, of course, the discussions I'm having the last year, the last 2 years, have completely changed. I've been especially busy with preparedness and crisis response, strategic stock, CBRN, NATO preparedness also. And so we clearly shifted to a more EU strategy, and especially to do things more independently from other continents, from other neighboring countries. And so it's not only about the EU, it's also in part, let's say, about ethics. Are we that convinced that AI is always a way forward? And I enjoyed really the discussion on, okay, what about consent? And even if you use consent, all the hurdles you still encounter. So at level of WHO, increasingly we have those discussions, and of course WHO is not putting forward any legal, let's say, boundaries, but especially at level of ethics and normative discussions, I think WHO has a large, huge added value, although as you have seen, some modern countries have stepped back from WHO, unfortunately. And of course, OECD is also always a little, let's say, a little bit behind maybe for us, but again, in general, is quite important at the level of healthcare systems and healthcare management. I'm not going to go into all the details. It's way too late this afternoon. European Health Data Space has already been mentioned. I don't think that, especially not in the healthcare sector, but also at the level of citizens, we already know what's in there. The AI Act also, I'm quite happy and proud that Europe already has an AI Act. At least we have a starting point. And a lot of questions, of course, on the implementation that will happen. And now the digital omnibus, I think, is also interesting to see, can we put some focus on it and really forward some sectors, some flagships in this interesting developments. So what about governments? Shouldn't they step back? Let entrepreneurship and research prevail? I think in part we should do. And not to hassle too much, not to impose too much administrative burdens, and we're quite good at that. But at the same time, of course, it's up to governments, I think, to guarantee certain principles, to implement or at least to safeguard certain societal values, and especially also at this point, I think, to guarantee quality, clinical efficacy, but also patient safety at the moment that you start applying these technologies to patients. So it's about benefits, it's about risks. It will also be about money. because the business models also will change. So the key messages, next to a major thank you all for these very interesting presentations, but also to all participants and of course the organizers. I think that Europe is clearly defining this as one of their major strategic priorities, but also with clear emphasis on values, on ethics, so a specific EU strategy. And we do see that just the fact that we are collaborating, that we are developing a joint strategy as European Union is being criticized largely by other, let's say, political powers in the world. We should be aware of that. We do have a lot of, that's what Europe also does. China is ruled by engineers, I'm told. The EU is governed by lawyers. Maybe only one lawyer in the audience, so I must apologize. So we should be aware that indeed this also is not that easy to implement always. But again, the fact that we are having those discussions and starting to develop a number of legal frameworks, I think is something that we really do need for data and also AI. Belgium, I think we have a unique position. We're Belgium, but we're also European. Belgium is one of the founding fathers of the European Union. And we are quite proud of that, and we keep defending those EU principles. And so we try to be a bridge builder. We try to do much more at the European level than you expect from a small country like ours. I think specifically in this field, we have a lot to defend. It's about collaboration, but at the same time also positioning the unique ecosystem of Belgium with industry, the research community, and as much as possible, indeed, a supportive eco-culture, to put that one step further and try to keep Belgium at least, let's say, one of the forerunners. So, at the same time, it's a call to action. And so, if I have discussions with politicians, ministers, of course, they're always interested in AI, but okay, that was interesting, next subject. I keep emphasizing the time is now. Now you need to build the framework. Now you need to emphasize what we want to do to the ecosystem, and especially have dialogue with your stakeholders on this issue. What do we need? Hold on, excuse me. Yeah, so the last part is also maybe the interesting part. I think that maybe you're not aware of that. If you do European benchmark on the use and implementation of electronic health records, digital healthcare, Belgium is really at the top. Like we were during the COVID vaccination campaign, but that's a different subject. So we should be proud of that. Although in reality, of course, in the hospitals, it's not always that easy. We do know that. So that's a starting point. That's quite well. So let's stress that, be proud of that, and use it. I think that at the level of EHDS, there were also some Belgians who are really in front or really in the middle of those European negotiations, which we should be. And we even have some European countries where they still have the medical files on paper, which is unthinkable, I think, in our system. And we have our Health Data Agency, which was founded in the meanwhile about 2 years ago. And I see that they're really evolving, maybe too slowly for some people. But again, the basics are there. And I think we're really moving towards a momentum that they will indeed provide much larger accessibility, data access than we ever seen before. Because we do know at this time point, a lot of data sources are being protected, not only in hospitals, also some other partners in our healthcare system who have a lot of data. So that's the next step, and I think with the Health Aid Agency and legislation, we already have the building blocks are there. So it's up to you, it's up to us, it's up to me to some extent to do that and to be ambitious, but also to deliver, I think. Thank you very much.
Thank you, Mr. Amakats. I don't know if some of you have questions or comments. No? Otherwise, I think that today it is the last session of the AI week, right? And I think there are some refreshments. Forstin, you want to say something? Or no? Well, yeah. So.
You can say the final word. We have a cocktail.
Yes, indeed. So work hard, play hard. Let's meet around the drink.